Okay, so this is the lab review for Lab 10. So we're going to be going over the thalamus and all of its different nuclei and some of the little grooves and indentations surrounding it. So to start, your thalamus is going to be right here in this Heimer dissection. You've got the top of the brain taken off, so you can see both of your thalamuses right here. And the thalamus is going to lie medial and inferior to the caudate nucleus. So therefore, this is the caudate nucleus. You've got your head, body, and you can tell you can't see. And where these are separate, so where the thalamus ends and the caudate nucleus begins, you're going to have this indentation. And these borders are called your thalamostriate grooves. Aptly named because in this groove your thalamostriate vein exists, which you can really see it here. And your thalamostriate vein is going to lie deep in that groove to your stria terminalis, which are axon fibers just traveling to deliver some information. So both of those are going to be in your thalamostriate grooves. And then, so that's your lateral thalamus little tract that you have to keep track of. Then you have a medial one that you can keep track of as well, and it's called your stria medullaris thalami. And so this is going to be kind of below this structure. It's going to be right on the medial surface of the thalamus, so a little bit deep to this. And that's going to be going back towards your habenula, which are going to be on either side of your pineal gland, which you'll see a better picture of in a second. So yes, so here is just sort of a gross specimen looking from the posterior, because here's your quadrigeminal plate or your tectum, and then here's your pineal gland. So here are your thalamuses, and here's your caudate nucleus. So again, here's your thalamostriate grooves right here. You can actually also see the massa intermedia, or the interthalamic adhesion, where these are communicating. And above the level of this adhesion, and you can see it arching right here, is going to be that stria medullaris thalami. And so these are going to be on the medial surface of the thalamus, and what we'll see in a second is that medial surface is going to be occupied by the dorsomedial nucleus of the thalamus. So that's what this stria is touching on. And that's going to come back, and they're both going to end up in these habenula right here, which are two nuclei. And they're going to sit on either side, like I said, of the pineal gland right here. Oh, and also, you see here this little outcropping of the thalamus, a little bump. These are going to be your anterior tubercles. So it's where your anterior nucleus are sitting deep and punching out this little blob at the anterior portion of each thalamus, but we'll see that in a few different cross sections and such. So first, I'm going to dive into sort of a mid-level cross-section of the brain, a coronal section. So we're at the level of the mammillary bodies. And this is important. It's going to be more important later when we're talking about distinguishing thalamic nuclei. But for now, just look here, see the mammillary bodies. You're going to see this massa intermedia, the interthalamic adhesion connecting the two thalamuses, or the thalami. I feel weird saying that. And then, once we pointed out on the previous slide, the stria medullaris thalami sitting on the medial surface of the thalamus, those are a bundle of axon fibers, and this is an axon stain. So those are going to be dark. So you can actually see right here on the medial surface of the thalamus the stria medullaris thalami. And also, I had pointed out the thalamostriate groove as being the border between the thalamus and the caudate nucleus. So we remember here that the lateral walls of our lateral ventricles are our caudate nucleus's body and head. And here's our thalamus. So then in between these two, we're going to have our thalamostriate groove area right here. And you can't really see the lumens of the thalamostriate veins quite yet, unless this could be the lumen sort of opened up. But we'll see that in a few other cross sections. So this area is going to be where your thalamostriate groove, that vein, and your stria terminalis is going to be located. And then just a look ahead before we start really learning how to ID the different thalamic nuclei. At the level of the mammillary bodies in cross section, this most superior nucleus up top is going to be your dorsolateral thalamic nucleus. Anything up more anterior in cross-section, anything like further forward from the mammillary bodies, this area is going to actually be occupied by your anterior nucleus, which is why you saw the anterior tubercle earlier. But right now, at here and behind, this is going to be the dorsolateral nucleus. And we'd already said this is the dorsomedial. And then coming out more laterally, you're going to have some of your lateral division nuclei. Up here, I'll explain why later. This is going to be your VL nucleus. And then down here is going to be your VPL and VPM. They're indistinguishable at this point. Okay, so now we're going to go to a mid-sagittal cut of the brain, which you've seen a million times. First thing, I'm just going to distinguish the regions of the thalamus, hypothalamus, and epithalamus. So your thalamus is right here, and you can actually get a pretty good idea of the shape sitting right there. Below that is going to be your hypothalamus, so that makes sense. It's going to include your infundibulum, optic chiasm area, anterior commissure, this part. And you can see actually pretty well this groove right here is separating your thalamus from your hypothalamus, and that groove is your hypothalamic sulcus. And that's going to be on the wall of your third ventricle right there, your hypothalamic sulcus. And then this is also just a good way to point out all of the recesses. So recesses are just any little divot that's sort of going inward or deep that you can see, and they're pretty well named, literally just sitting right next to these structures. So your pineal recess, if you find your pineal gland, which is here, here's your posterior commissure. This little like valley that's leading into it, that's your pineal recess. Then you can look over and find your optic chiasm, this little recess leading into that, that's your optic recess. And then your infundibular stalk, where your pituitary was right here, like here's where it would come down into your pituitary gland. It's been cut, obviously, but this recess leading into that hypothalamic stalk is going to be, or your infundibular stalk, sorry, is going to be your infundibular recess. Okay, and we can see here on this cross-section, you got your mammillary bodies here. You've got this anterior-most border of your hypothalamus, your lamina terminalis, that anterior wall of the neural tube, per Dr. Smith. And like I said, also your anterior commissure is going to be right there. Your tuber cinereum is a bump that isn't super easily appreciated, so it's more you need to just know the location of this. The tuber cinereum is going to be right posterior to the optic chiasm, and it's going to be continuous with the infundibular stalk. So on this cross-section, it looks like it's going to be about right here. The way you would see this bump the best is on the bottom of the brain. If you flip it over, you can see a little bump, but it's going to be right in this location. Okay, so now we're going to get into some cross-sections and just sort of see those structures in a different way. So as I had said, the Thalamostriate groove, you're going to find between your caudate, which you can barely see it anymore, your caudate and your thalamus. So this area is going to be your Thalamostriate groove. You can see your little vein lumen right there. And then the black axon fibers, that's going to be your stria terminalis. And then your habenula, I said earlier, that is what's coming to an end when your stria medullaris thalami are going to go anterior to posterior, and they're going to end up at these habenula that are on the lateral borders of the pineal gland. So they end a little bit before the pineal gland really gets into its full glory, and you'll see that. You've seen that in the gross specimens I showed you earlier. They kind of have, your two, and then your pineal gland sort of sits a little bit more posterior. So you can see that really well here. You can see your pineal gland just now starting to show up in this cross-section. And if that's your pineal gland, then this area is your superior cistern, because your pineal gland projects into that superior cistern. So no longer third ventricle. And then here you can see your two habenula. And so this is the end of that stria medullaris thalami, and as this is your dorsomedial nucleus of your thalamus, so that all makes sense. And then this is also a really good image of your fasciculus retroflexus. So what happens at your habenula, the nuclei, they are going to communicate with interpeduncular nuclei. And to do that, they have this fasciculus retroflexus, which is easier to say, but what makes more sense is the name it's given is the habinula, interpeduncular fasciculus, and that's what's connecting the two. And you can see this here with the myelin stain. So here's your habenula, and here these black pathways are your fasciculus retroflexus headed down to those interpeduncular nuclei. And let's see, and the other thalamic nuclei we can see here is your lateral posterior nucleus. It's going to be these right here, but don't worry too much about that. We're going to get into how to ID these later. Let's zoom in on this area so we can get a better look at what nuclei we're looking at. Okay, so we're going to talk about these in the order of which they come, anterior to posterior, but just for a quick little sneak peek, we've got our lateral posterior nucleus, and this is going to be sitting right anterior to the pulvinar. So you can see we're in the midbrain, so we're pretty far back in our brain cross sections, but we don't really see the posterior commissure yet, but we see the red nuclei. So we're pretty far back, but not quite far back enough that we see a really good thick pineal gland. And it's that cross section where this would be your pulvinar, because that would be the most posterior part of your thalamus. But we're right almost there. So this is going to be your lateral posterior nucleus. We still have your dorsomedial nucleus heading up that medial side of your thalamus. And then down here, we get a good look at some of your ventral tier nuclei. So, and you can actually see them separated pretty well here. So you have your VPL and your VPM. And then you also have a good look at your intralaminar nuclei, which are your centromedian and parafascicular. So your centromedian is here. You can see it surrounded by that lamina. And then on this side is really the only part you can really see it well separated. This is your parafascicular nucleus, just this little part that almost looks like it's a big part of this intralaminar nuclear group. Right there. Okay, so here's a quick slide reviewing the internal capsule. And I've got 3 different views as you go further back in the brain. So let's start with this overview specimen. We're looking over the top of the brain down onto it. So this is a myelin stain, so the black is going to be the internal capsule. You have your anterior limb, your genu bending right there, and then your posterior limb. And then right here is going to be your retrolenticular, because here's your lenticular nucleus, or your lentiform nucleus, which is your putamen and your globus pallidus. When you can't really separate them out, they make a lentiform nucleus. So retro behind lenticular, this area right here. Now looking over at cross sections, here's how you're going to separate the limbs and the genu in coronal sections. So here you can tell you're pretty anterior because you have the nucleus accumbens. That's where your caudate and your putamen are actually merging ventrally. So you're pretty ventral. That said, when you see the internal capsule, that's going to be your anterior limb because you're so far anterior. Then you move a little bit further back in the brain. You have your anterior commissure, so you're still pretty anterior. But now you've got your foramen of Monroe. So now you can tell you're a little bit further back. And if you were cut a little bit behind this, you would start to see your anterior nucleus of your thalamus creeping in, which is right here. But we can't even quite see that yet. But this location where we're at the foramen of Monroe, we're right at the anterior nucleus of the thalamus, that's going to be your genu of your internal capsule, so right here. And you can tell here because here's your foramen of Monroe. So you cut through that, you're right at the genu. Now let's move a little further back. Now once you see the thalamus really bust in, and your thalamus is sort of pushing this internal capsule out, that's going to be your posterior limb. And then your sublenticular, you can't see very well on these cross sections, but really it's going to be your axons, your internal capsule underneath of your lentiform nucleus, your putamen and your globus pallidus. So like these axons could be a part of, of that sublenticular part. We'll see another stain of it in a few slides, but it's more important that you just know what runs through these parts. So the important things off the top of my head Your anterior limb is going to be your frontopontine fibers. You're also going to have communication from the anterior thalamic nucleus going to the cortex. And then your genu, that's where you're going to have your corticobulbar fibers. And it's also specifically somatotopically, you're going to have your fibers going to your face right at the genu. Then your posterior limb, you're going to have all your descending corticofibers, so going down to your spinal nerves and things like that. That's the somatotopic organization where it's head up here, going down to your feet down here. Your retrolenticular part is going to have some of your superior retinal field axons coming back here. And then your sublenticular part specifically, there's a couple things we want to know that's in there. You've got auditory radiations, and you also have your inferior retinal field fibers traveling through there. So here is the thalamus. It's really, the best way to do it is just really try and get a good 3D image in your head of this and sort of imagining this cut any which way, which ones are you going to see if you have this cross section. So this is just for you to review all the functions and such, and you can come back to this for a guide when you're looking at these cross sections and wondering how you could be seeing what you're seeing. So big takeaways before we go in to start IDing. This is anterior, this is posterior. So the first things you're going to hit, you're going to see your anterior nucleus on top. Anterior nucleus on top, your anterior tubercle you're going to see. And your most anterior thing you're going to see next, you're going to see your VA, which a lot of the times it's cut sort of right here, so it's going to be VA/VL. And then you're going to see your dorsomedial nucleus on the medial surface pretty much the whole way, as you can see. So pretty much that whole medial section is going to be dorsomedial nucleus. Once you get a little bit far back, like once you start seeing your dorsolateral, once you get to the mammillary bodies, see, like this would be the cut where the mammillary bodies are, because it's not anterior nucleus anymore. It's going to be dorsolateral nucleus. And at that point, you're going to see probably that your lateral side is going to be your VL, medial side still being your dorsomedial. And even still, as you get a little bit further, you may still see some of that dorsolateral on top, and then you're going to start seeing your VPLs and VPMs down below, maybe even a little bit of your VL left above those. And then, as I was saying earlier, right before you get to the true back of the brain cross sections, like you can still see the red nucleus, but we haven't gotten to the posterior commissure yet. You may see the lateral posterior nucleus instead of the pulvinar, and they're going to sort of be in the same looking location. This is sort of poorly drawn because you can't really draw inside of this, but the centromedian is going to be really like deep and in here, so you're going to be able to see it in the middle when you cut a cross section. And then obviously your LGN and MGN we've identified plenty of times before. So let's just take a look. We're going to start anterior. So this cross section is actually a little more anterior than this one, and that's why I wanted to show you that there's two different sort of cross-sectional locations where you can see your anterior nucleus pretty well. So here we have this is like you've just cut into the thalamus. You are just far back enough that you just cut the tip of it off. So all you can really see here that's the thalamus is going to be your anterior nucleus. And you can see too that really big bump coming out into that lateral ventricle. That's going to be that anterior tubercle we saw on the gross specimens earlier. And then also I'd like to point out here, this is, your thalamus just starting to show. And we know that your hypothalamus is obviously deep, and inferior, rather, to your thalamus. So this little divot is actually going to be that hypothalamic sulcus that I pointed out on the mid-sagittal cut. And then if you look over here, we're going to go a little bit further back, but we're still pretty anterior. You've got your anterior nuclei sitting on the top middle like that. Here you've got a good look at your dorsomedial nucleus. And then this lateral part, since we're still pretty anterior, is going to be your VA leading into your VL. It's kind of hard to discriminate what they're going to be about this part. You're either going to have your whole VA with a little bit of your VL starting to show. So just know this is your VA-VL area. And then also, this is a fun little tidbit, is when we're studying the thalamic nuclei, we learn that the anterior nucleus is going to be receiving input from the hippocampus and from the mammillary bodies. And so the mammillary bodies are going to communicate to the anterior nucleus via the mammillothalamic tract. And you can actually see this dark stained right here. This is the mammillothalamic tract actually leading up to go to this anterior nucleus right here. Okay, so now we're a little bit further back. You can see we've reached the mammillary bodies. The mammillary bodies, that should be your alarm bell that we are no longer seeing the anterior nucleus. We are seeing the dorsolateral nucleus. So these little ones up here, although they look like they were the anterior, we know that we're far back enough that we have gone back enough on that diagram to hit the dorsolateral. We still have your dorsomedial right here in the middle. And you see that stria medullaris thalami right here. Here's your thalamostriate vein, your thalamostriate groove as a total, as a whole, and then your stria terminalis is some of this myelin right here, the axon fibers. And then when we go laterally, we're a little bit further back now, so we can see that we are at the VL level. So whereas a little bit further up, we were like, okay, we're in the VA, VL area. Now we are in solid VL territory, right in the middle of that thalamus. And then you can see that a little bit below in this ventral tier, we're going to start seeing the VPL, VPM area. So at this point, they're pretty indistinguishable. So just VPL, VPM. And then when we get a little bit further back, we're going to see separation between the VPL and the VPM. But it's really good to just look at each of these cross sections and maybe as I'm explaining, go back and look at that diagram that shows you the whole thalamus in 3D space and sort of see how, you know, for example, you could see your VL and your VPM, VPL at the same time. Okay. All right, so now we are at that image that I showed before where I was like, okay, we're at the midbrain. We can clearly see we got the big red nuclei. We can see your pineal gland, your habenula, but we're not quite all the way posterior. Otherwise, we would see, you know, a good posterior commissure. So for that reason, we are definitely far back, but not all the way far back to where we could say this is the pulvinar. So this is just before the pulvinar, which is going to be your lateral posterior nucleus. We still have your dorsomedial, old reliable, is going to be right here. And this just goes to hit home the fact that your stria medullaris thalami was going to be running back that whole surface and ending on your habenula right here. And now we can see those VPL and VPM have a good separation, right? And then we have this one in the middle, and we're like, okay, what is that? Well, we have intralaminar nuclei, and these are the ones that I was saying are just sort of deep into the thalamus. So this big one that you can see most easily, this is your centromedian nucleus. Makes sense? Central in the center, median in the middle, what have you. Now, there's also a parafascicular nucleus. Both of these together are intralaminar nuclei. Your parafascicular nucleus is a little bit harder to point out, but you can see it pretty well on this right side. This is your parafascicular nucleus, but together they are intralaminar set of nuclei. So parafascicular, centromedian. And then a good way to remember your VPL and VPM are down here is that Dr. Smith says that your VPM, which you can see right here especially well also, is going to be, it can be somewhat crescent shaped. It's not quite so right here, but it crescent shapes and will hug the bottom lateral part of your centromedian nucleus. So you can see your VPM sort of hitting up against that border of your centromedian nucleus. And then, just to point this out, so I had said earlier that since this is the pineal gland, we know that this is the superior cistern that it's projecting into. We know that these lumen right here are actually going to be your internal cerebral veins, because these are what have been made up from your thalamostriate veins, your septal veins, which I don't see a lumen right here in your septum, but it would be in your septum pellucidum anyways. And then your anterior choroidal veins. The three of those come together and make your internal cerebral veins that are going to course anterior to posterior right in the superior cistern. That's where they're going to eventually become the great vein of Galen. Okay, so now this is what I mean by we're posterior proper. We're like, we can tell we're really posterior because we've got a good posterior commissure right here. The pineal gland is way more evident, it's less fuzzy, it's more resolute. And you can see that you've pretty much just got one big old thing left, right? That's going to be your pulvinar. So that's your part of your visual extrageniculate pathway. And then down here, you can see those two little things or if you're like me, didn't really even know were part of the thalamus until recently. Your median geniculate nucleus and your lateral geniculate nucleus. And you can see them on both sides here. So medial and lateral. And then just for your information, if you see some black, some myelin axons, and they're leading and going into this LGN, that's probably your optic tract that we learned from the last exam. And then this is also just a little look forward, a little sneak peek to next lab. We're going to be learning a lot about the hippocampus. And we should have drilled home at this point that your caudate is going to be your lateral border of your lateral ventricle. And then your tail of your caudate, so it's going to arch like a C backwards and come forward, and your tail of your caudate is going to be your roof of your temporal horn of your lateral ventricle. So just like your lateral ventricle is a C that sort of C's back and comes forward as this, your caudate's going to do the same and become the roof. Well, the floor of the temporal horn is your hippocampus. So we're going to learn there's a few different parts of the hippocampus. I believe this is your subiculum, hippocampus proper, and then your dentate gyrus. This is just your hippocampal formation, and that's going to be your floor of your temporal horn of the lateral ventricle. And then last but not least, just wanted to point out the internal medullary lamina on a couple slides. The internal medullary lamina is what's going to be separating out your anterior division from your lateral division and medial division of your thalamic nuclei. So if you look over top, it's going to be Y-shaped. So here's your thalamus, and then here's your internal medullary lamina, and here's going to Y, and the Y's are the little Y arms are going to be enveloping that anterior nucleus, a little bit of anterior tubercle you could see on the gross specimen. And then here's your lateral division and your medial division. And then you can also see it on cross-section at some points. Here is that clear lamina. And again, mammillary bodies, this is going to be your dorsolateral, not your anterior nucleus. But yeah, so that's your lamina. That is your thalamus overview. Any questions, you can find me, talk to me, ask me at the lab review, but that is going to be it.